Hi again, everybody. It's Bill Cavanaugh from abscopal where we're going to keep talking about the history and the current state of the abscopal effect as it relates to the treatment of cancer. We're going to keep talking about it for reasons that I'll make, and I hope I'll make clear. And we're going to keep talking about it until we get some serious traction for real research into this topic. We've talked a little bit about history. We've talked about Robert Moll. We've talked about Dick Ablin. We want to talk about cases and get more specific. But before we go any further, we need to talk just a bit about cancer biology. Now, that might sound daunting, but I promise to keep this simple and boil it down to just a few points. Clip the treetops, if you will. Now, you're not going to be able to pass any exams after this video, but I think you will be able to start seeing why the abscopal effect is being witnessed more and more often. What we want to do is make it happen even more often. The problem is that this topic, cancer, why it happens, so forth, just keeps getting deeper and deeper all the time. But there are a few conclusions that I think are unavoidable and that will help our documentation of abscopal effects make sense. We're going to kick off episode 3 with some facts to try to frame this whole problem, explain why we're doing what we're doing. These are numbers from the American Cancer Society for 2026. All right, this year, somewhere in the neighborhood of 2 million new cancer patients will be diagnosed. These numbers do not include non-melanoma skin cancers either. They will add to the 18 or so million who are already living with cancer in this country. And of the 2 million or so cancer patients to be diagnosed in 2026, roughly a third will be dealing with cancer that is not localized. Non-localized meaning cancer that has significantly invaded outside of its original location or has spread either to the lymph nodes in the area or to another organ. Here's an image of our hypothetical patient, we'll see again and again, showing what I think everyone understands intuitively. In the case of localized cancer, as in the colon cancer patients shown on the left, the chance of cure is reasonably good, surgery being traditionally the gold standard for cure. But once the cancer is spread, such as in the hypothetical patient on the right, where the primary cancer is metastasized to the lungs and liver, cure becomes much less likely. And unfortunately, over 500,000 people will die with their cancers this year in the US, the vast majority with metastatic cancer. Given the number of family members and friends and colleagues of these patients, it seems that everyone will somehow be touched by cancer in this country in the year 2026. While progress has been made in treating advanced cancer, that is, cancer that has grown beyond being localized and treatable by surgery, we still have a long way to go, judging by just how many people are still dying. That's going to be the focus of what we talk about here at abscopalevents.com. Bear with me if you can. The objective and the evidence will become clear as we go on. But we cannot go much further without going over some basic cancer biology. I promise to make this as painless as I can. Let's start with the standard question. What is cancer? As you've probably heard, cancer is actually a number of different conditions, primarily defined by the problematic cell type or of origin, of which there are many. But for our purposes, we are going to restrict our conversation going forward primarily to cancers of epithelial origin, or carcinomas, as they are known. Because, first, carcinomas make up more than 80% of all cancers, and second, as a group, they bear certain similarities. Carcinomas and adenocarcinomas include cancers of the colon, breast, lung, ovary, prostate, liver, bladder, and on and on. And Almost all of the abscopal effects that we will be reviewing here are going to be cases involving carcinomas. But for essentially all cancers, including all carcinomas, cancer cells give themselves away visually under the microscope. This is the reason why biopsies are taken when cancer is suspected. In fact, a cancer diagnosis can only be made when the biopsy or tissue sample is looked at under the microscope. There are microscopic giveaways for every cancer that lead to what's called a microscopic diagnosis. A classic illustration of the microscopic features of cancer is illustrated by the Gleason grading scale of prostate cancer. This only applies to prostate cancer, but the Gleason grading scale is seen here. As you can see, as we reach higher grades of cancer, as we go from 1 to 5 on the Gleason scale in this case, you can see what started as fairly uniform cells organized in clusters, gradually becoming more disorganized, misshapen, end up looking nothing like their benign predecessors, using grade 1 as essentially normal for the Gleason scale. And as we move from highly survivable prostate cancers in the grade 2 to 3 range under the microscope, we reach very dangerous and deadly cancers at grade 4 and 5. So in terms of what is cancer, number one, cancer looks like cancer under the light microscope and is identified or not as such by a pathologist. That's where the official diagnosis of cancer takes place. And secondly, cancer cells exhibit a set of behaviors that set them apart from normal or benign cells. These features include the fact that cancerous cells stop responding to normal cell signaling. One of the lessons of cell biology over the past half century has been the discovery of just how much communication there is among and between cells. And, of course, just how much cancer ignores this communication. Secondly, cancers continue to grow even when there is no need to, and they attempt a sort of immortality. While most epithelial cells reach a limit on the number of divisions they can make before undergoing a sort of planned cell death, carcinoma cells, at least certain of them, keep dividing without apparent limit. This is another strange behavioral change in our once docile epithelial cells that have transformed into carcinoma. Cancer cells also show a tendency to invade their neighbors' spaces when, especially with epithelium, these cells are supposed to stay put and respect boundaries. In fact, there's a whole set of rules that epithelial cells more or less obey until they don't and become carcinomas. And worst of all, cancerous cells show a tendency to mobilize, travel, and grow in environments far away from where they originated, in places in which they are not originally designed to grow. Given the appearance of these cells under the microscope and their strange behavior, you might wonder, are the typical twisted appearance of cancer cells under the microscope and their aggressive, destructive, and potentially deadly behavior related? Well, it certainly would be strange if they weren't, given that both have been associated together as hallmarks of cancer for well over 100 years. So the answer would appear to be yes. Now, in addition to the what is cancer question that I hope we've answered in general terms, there's also a why is cancer. From here, we're going to go with the dominant thinking these days, and that is that what we are dealing with in terms of cancer is one way or another related to mutations or mistakes or in undesirable elements in human DNA. For our purposes, we're going to call all of these problems DNA mutations. Okay, this is going to be our, as usual, highly simplified illustration of a DNA mutation. The colored lines that connect our familiar double DNA helices in our example are together, both of them together, called base pairs. Base pairs in the long runs contain the genetic code that I think we're all roughly acquainted with. In our left-hand example, the pairs are orderly and connected like they're supposed to be. But in the right picture, you see a set of base pairs broken apart. It is not supposed to be that way. This is a mutation in the DNA. Again, a very simplified example, but it'll serve our purposes. Now let's quickly look at one of the primary tenets of biology. The fact that the totality of our DNA, altogether called the genome, contains about 20,000 genes in round numbers. And those genes are instructions for about 20,000 proteins. Genes encode instructions for proteins. That's what genes are. And proteins are what make life happen at the cellular level, which allows life at the organism level. Proteins are the most functional of biomolecules and perhaps the most fascinating molecules of them all, period. But as a result of, we're just going to say mutated DNA, we can get, sort of by direct implication, proteins, protein products that contribute to the anaplastic or highly problematic behaviors of cancer that we just went over a few minutes ago. Here's one of the best known and most studied set of mutations in human DNA. Mutations of the DNA encoding the p53 tumor suppressor protein. This is a diagram of protein p53 that is coded for by the p53 gene. On the left is the normal p53 protein, which, when operating according to its normal conformation, actually works to prevent DNA mutation. Mutations in the p53 DNA lead to mistakes in the p53 protein itself, which are indicated by arrows inside our red circle in the right-hand image. These arrows point to specific parts of this important anti-cancer protein that are not as they are supposed to be because the DNA that held the instructions for this protein was corrupted or mutated. So mutated DNA, and I'm not talking about allelic changes over time that are inherited over generations that are not related to cancer. I'm not talking about those. We're talking about the kind of DNA mutations that can get us cancer cells and all the uninhibited growth and invasion and ultimately metastasis. Those mutations are the ones we're most concerned with. So, to recap, cancer is something that A, looks like cancer, and B, behaves like cancer. But the reasons that cancers exist is related to mutational burden that results in faulty proteins that get us cancer. But there is an increasingly compelling idea that mutations occur to DNA due to mistakes that occur in our genetic makeup and environmental surroundings and who knows what else, as a matter of course. In other words, mutations happen to our DNA, and this could be true over much of the lifetimes of most humans. These mutations, these insults, they occur. But whether they involve mutated receptors, disabled tumor suppressors, or any of a host of other defective and dangerous mutated proteins, somehow these mutations do not always create life-threatening cancers. You understand? Mutations happen throughout life, but somehow, for much of our lives, they don't create life-threatening cancers. Please bear with me, it's going to make sense. Now, especially in epithelial cells that divide quite a bit, there are errors made in the copied DNA. Cells are exposed to chemicals, UV radiation, all kinds of insults that can mutate DNA. But mutations in DNA do not seem to automatically lead to rogue cells operating with malignant effect. It is now appreciated that an immune system function known as immunosurveillance actually destroys cells displaying mutations before they can grow into full-blown cancers, carcinomas for the purposes of our discussion. And believe it or not, this hypothesis is becoming ever stronger of late. Our immune systems are filled with aggressive soldiers that clear invaders and parasites like bacteria and viruses. And in a similar way, these same elements of the immune system are thought to be entirely capable of clearing cells exhibiting mutated gene products or mutated proteins, as we have been talking about. And this process may happen over much of our early lives. So what happens that allows these lethal cancer cells to thrive when they should be confronted by killer immune systems bent on destroying them? This is a huge question. What happens? It turns out that through mechanisms that are still little understood, cancers that acquire malignant capabilities via DNA mutation and protein transformation are actually protected by elements of our immune system. This is one massive finding. The apparent fact that cancers that reach a metastatic stage are assisted by having recruited our immune systems into their malignant effort. As an example, and we've simplified here again, of course, here is a depiction of two kinds of T cell interactions with cancer cells. On the left, we have the interaction that we all thought was the standard T cell response to these rogue cells. Kill them all. But on the right, we have a newly appreciated interaction between T cells and cancerous cells. This interaction involves a sort of mirror image of the killer lymphocytes on the left. But rather than killers, they are protectors. Yes, these regulatory lymphocytes, which are intended to protect your normal tissues and organs against your killer T cell attacks, have been recruited by cancerous cells and duped into protecting them. In fact, the 2025 Nobel Prize in Physiology or Medicine went to 3 individuals who identified the hallmarks of these regulatory T lymphocytes. And there's another mechanism by which cancer cells trick killer T cells from destroying them. This one was the subject of the 2018 Nobel Prize in Physiology or Medicine and involves a direct disablement of killer T cells, of killer T lymphocytes, by the cancer directly disabling these killers by using a switch called programmed cell death protein 1, or just PD1. In this case, cancer cells have acquired the ability to press a button on our killer T lymphocytes in order to turn them from effective eradicators of cancer cells into mere bystanders. I think it's informative that 2 Nobel Prizes in Physiology or Medicine over the past 8 years have gone to discoveries related to mechanisms by which the immune system might stand down to cancer cells rather than destroy them. Which leads us to the emerging position that, at least in significant part, cancer is, in a way, a disease of immune dysfunction. I'm going to stop here on our cancer biology overview. That's a lot. There's lots more to talk about, and I will. But I think I've laid enough groundwork for everyone to start, I hope, to see why maybe we are seeing more mentions of the abscopal effect in the medical literature. Again, I'll come back to this slide. Here we go. I mean, we're just something we never heard of suddenly is being talked about more and more. So, quick review, and please keep in mind there's a lot of speculation on my part here. This is very little is absolutely provable. I'm just trying to put pieces together. But here's an overview. First, human cancers that acquire a malignant phenotype or set of dangerous abilities may well do so secondary to mutations in DNA. Mutated DNA makes mutated protein. Our immune system probably holds the ability to eradicate these mutated cells with their malignant abilities, but our immune system, in these cases, has been declawed by the cancer itself. Now, we can't do much at the current time about the regulatory T cell problem. We can do something about the PD-1 problem, the second one we talked about, using monoclonal antibodies that have been developed just for this therapeutic purposes. These are PD-1 inhibitors that we have been calling, even the last couple episodes, immunotherapy or cancer immunotherapy. So what we're going to do is look at a case, review a case. That's what we're going to be doing a lot more of after we get through a lot of this groundwork. But this case was published just 2 years ago. And please keep in mind, much of what I will be reviewing in these videos are case reports. They are not clinical studies that are capable of proving anything. But, as I will keep saying, we may be witnessing the beginning of something important in cancer treatment. And this something important will likely not engender large-scale study for reasons I will talk about in another episode. Okay, let's look at this case that involves an abscopal effect, fascinating case, in the treatment of carcinoma of the liver or hepatocellular carcinoma. This case is reported out of the Mayo Clinic in Jacksonville, Florida, where a 43-year-old male patient with advanced liver disease that included cirrhosis and hepatocellular carcinoma was seen by the physicians there. This patient was badly in need of a liver transplant, but because the liver cancer was extensive in his case, both in terms of tumor size and number of tumors, he did not meet the criteria for transplantation. In these cases, there can be an attempt to downstage, as they say, the liver cancer, or reduce the number and or size of the liver tumors in order to fit this patient back into the criteria that govern the suitability for liver transplantation. There are numerous ways to reduce the size and number of liver tumors. They usually involve putting a probe of some sort into the liver tumor. You can do right through the skin, right through the abdominal wall, in an attempt to cook them or freeze them or somehow or another disable or destroy one of these liver cancers. There's another means by which material, sometimes radioactive, sometimes it's chemotherapy, sometimes it's just nothing, can also be introduced into the vessels of the liver in order to break or destroy the vasculature, the blood supply to these tumors. Oftentimes, any of these can be used to diminish these tumors, to downstage people for liver transplantation. In this case of this young man that we're discussing at Mayo Jacksonville, several efforts over 2 years, many even, had failed to impact these liver tumors, including the things I just described, sufficiently to downstage him into eligibility for transplantation. Now, here's a schematic drawing based on what the authors in this paper reported. The hepatocellular carcinomas appear in red based on imaging studies that the authors reported. You can appreciate 2 large tumors and 4 smaller ones spread throughout the liver. With this extent of disease, liver transplantation was off the table. After numerous unsuccessful attempts to impact the disease, the patient was started on immunotherapy using a PD-1 inhibitor, such as the ones we've been discussing and calling cancer immunotherapy. But there was no significant response to immunotherapy by itself. His physicians then decided to freeze two of the larger tumors in order to, as they put it, augment an immune response by means of antigen presentation and local inflammation. So they're trying really to provoke an immune response along with this anti-PD-1 immunotherapy. Fascinating, right? Because by freezing, we talked about last time, the contents of the cancer cells are expelled, and this would include any mutated protein, any other material that might be contributing to the malignant and very difficult to treat nature of these liver tumors. Here's the schematic therapeutic freezing plan that was undertaken. The patient remained on anti-PD-1 immunotherapy over the time of freezing, before and after. Within months, the liver cancers in this patient underwent an apparent therapeutic effect as judged by blood test for liver cancer markers. The alpha-fetoprotein dropped significantly. And at 4 years after his cryotherapy, at 5 years following the cryo plus immunotherapy, he underwent a successful liver transplantation. And at the time of the writing of this case, he had no sign of cancer. Now, what makes the account of this patient so interesting was just how many attempts were made to downstage this young patient's extensive liver cancer, and the fact that his physicians very intentionally attempted to induce an immune response in combination with the anti-PD-1 immunotherapy with the cryotherapy when there just weren't that many treatment options left. They literally called the result abscopal downstaging of hepatocellular carcinoma. Hats off to them. Now, we will have more cases to talk about next time. I felt that we needed to get some of these biology concepts out of the way, because we will come back to them and mention them as sort of connecting the dots together. And I hope I didn't go on too long. I hope it wasn't too much of a challenge. Thanks. I will be back. See you next time.